Okay, now we were just looking at this picture of replication, and yeah, we were just looking at this picture of replication. Now, we're going through the different enzymes. But let me show you the picture we made on our board. If you can draw this, then you pretty much got it. But yeah, just kind of randomly labeled one side 5 to 3, which means the new strand is built towards that bubble, 5 to 3. Yeah, now if this was a picture of transcription, it would just be this one piece here. So if we were to transcribe RNA, the DNA would unravel, the RNA would be built here, and so this little green piece, this new strand, this what is the leading strand on DNA, if it were made with ribose, it would be RNA, and it could float around and get out of the nucleus and out into the cell. So once again, the lagging strands work away from that replication fork, away from that bubble. And then, yeah, we also asked you to understand the different enzymes that are involved. And really, once again, just think of them as classes of enzymes: helicase, primase, polymerases, ligases, isomerases, or a lot of times they're called topo or topoisomerases. Yeah, and here's that picture from the enzymes from 1982, all right, and we had started to identify them. Look how big these things are. The molecular weight is 900,000. Are you kidding me? I mean, it's really just kind of an estimate. We really don't know exactly how big or exactly what that structure is. But yeah, here's a lot of the proteins that enzymes we found that need to be there in order for replication to happen. All right, so replication happening in the nucleus, transcription happening in the nucleus. All right, and really it's pretty much the same. As far as our purposes are concerned, it's just a different set of enzymes, different set of polymerase enzymes. Instead of the whole genome, it's a smaller single strand. And you don't get thousands of bubbles opened up simultaneously everywhere. You just get the one little bubble and it closes at the end after you've made that RNA. But now there's more to it though, because after it's transcribed, here in this picture they call it pre-mRNA. A lot of books will call it the primary transcript, and then it gets processed and really stuff chopped up, a couple things added to the end, and now you have a mature RNA. But that's still that RNA processing, still part of transcription, really kind of happens in the nucleus still. So yes, if we look at slide 25, here's another picture. We went through this in an earlier screencast. But yes, you have your DNA, there it is, the primary transcript, there it is, it's cut, it's cleaved, stuff is removed, it's spliced back together. And then once again at the 3' end there's a lot of adenine. At the 5' end, this cap is a lot of guanine, a lot of GTP. Now keep in mind this whole processing and cleaving and chopping and shortening of, so from the primary transcript to the mature mRNA, it's a lot smaller. That only happens in eukaryotes, plants, animals, not happening in bacteria. All right, which is a good thing. That's really what allows genetic engineering to occur. We can feed bacteria DNA. We can feed and it will make the RNA that we want. It won't process it, it won't change it, and it will use it based on the DNA that we give it, that we feed it. All right, here's just, once again, more the same kind of pictures. Here's a piece of DNA with exons and introns. It's transcribed. It has the cap put on the 5' end, the poly A tail, the adenine put on the 3' end. That's the guanine, the GTP cap. The exons are kept, the introns are removed. So the splicing is that removing of the introns,